Good afternoon, ladies and gentlemen. It's my pleasure to talk on this topic, assessment of organ damage in SARS-CoV-2 infection with imaging modalities. I never thought of speaking on this topic, but the rapid spread of COVID-19 pandemic all over the world provoked me to collect possible information on this topic and share it with you. I am going to present 23 slides and it'll describe general information on source of SARS-CoV-2, diagnostic test available, drug development to treat infection, and 13 cases where PET, CT, and MRI discovered organ damage in an asymptomatic and symptomatic patient. Starting in December 2019, a cluster of unexplained pneumonia cases was reported in the city of Wuhan, the capital of Hubei province in central China. In January 2020, the pathogen causing this bilateral interstitial pneumonia was identified through high-throughput sequencing of the viral RNA genome. The World Health Organization, WHO, named this new virus Severe Acute Respiratory Syndrome Coronavirus 2, SARS-CoV-2, which belongs to the beta coronavirus cluster, which also comprises the viruses causing SARS and MERS-2. The consequent SARS-CoV-2 related disease was defined as coronavirus disease 2019, COVID-19. Coronaviruses inducing the outbreak of severe acute respiratory syndrome, SARS-CoV, and Middle East respiratory syndrome, MERS-CoV, were originally bat viruses that spread to an intermediate animal, civet cat and camel, respectively, which then exposed humans to the viruses. Genetic analysis of the coronavirus causing the novel COVID-19 outbreak, SARS-CoV-2, recently showed that their closest genetic relatives appear to be bat coronaviruses, with the role of intermediate species possibly played by the pangolin, although with some conflicting results. Recent studies shown that some bat coronaviruses can infect human cells without passing through an intermediate host. There are also rumors and suspicious guess that coronavirus isolated from bat, genetically modified or unmodified escaped from the virology laboratory in Wuhan. The lifespan of SARS-2 varies from 3 hours to 7 days and highly contagious. The researchers compared the SARS-2 virus lifespan on surfaces to that of the SARS coronavirus in a 21 degrees Celsius room at 40% relative humidity. They found that both coronaviruses, SARS and SARS-2, lived the longest on stainless steel and polypropylene, a type of plastic used in everything from storage containers to toys. Both viruses lasted up to 3 days on plastic, and the SARS-2 lasted up to 3 days on steel. On cardboard, SARS-2 lasted 3 times longer than SARS did, 24 hours compared to 8 hours. Temperature and humidity play a big role in how long the virus can survive. Increase in temperature from 21 degrees Celsius to 30 degrees, lifespan on steel surface decreased by at least half. Increase in relative humidity, high moisture in the air and mugginess makes it challenging for viral droplets to survive in the air and spread between people. With this science, the coronavirus cases in India should decrease in monsoon season. Early reports suggest the most common symptoms in SARS-2 infection are fever, 88%, and dry cough, 67.7%, which are shared with many other viral syndromes. Conspicuously, rhinorrhea, 4.8%, and gastrointestinal symptoms, diarrhea 4% to 14%, nausea or emesis 5%, appear to be infrequent in COVID-19. SARS-CoV-2, a single-stranded RNA-enveloped virus, targets cells through the viral structural spike, S, protein that binds to the angiotensin-converting enzyme 2, ACE2, receptor. Following receptor binding, the virus particle uses host cell receptors and endosomes to enter cells. A host type 2 transmembrane serine protease, TMPRSS2, facilitates cell entry via the S protein. Once inside the cell, viral polyproteins are synthesized that encode for the replicase transcriptase complex. The virus then synthesizes RNA via its RNA-dependent RNA polymerase. Structural proteins are synthesized, leading to completion of assembly and release of viral particles. These viral life cycle steps provide potential targets for drug therapy. Promising drug targets include non-structural proteins, 3-chymotrypsin-like protease, papain-like protease, RNA-dependent RNA polymerase, which share homology with other novel coronaviruses. Additional drug targets include viral entry and immune regulation pathways. No proven effective therapies for this virus currently exist. The rapidly expanding knowledge regarding SARS-CoV-2 virology provides a significant number of potential drug targets. The most promising therapy is remdesivir. Remdesivir has potent in vitro activity against SARS-CoV-2, but it is not US FDA approved and currently is being tested in ongoing randomized trials. There are 3 types of COVID-19 specific diagnostic tests. Number 1, molecular test. Molecular tests detect genetic material from the virus. Samples are collected on nasal or throat swab. A series of chemical reaction that amplifies genetic material of virus. High-intensity signal shows person is infected with SARS-2. Testing is carried at the hospitals and community clinics. The test is useful to isolate those infected so treatment can be provided and other potential cases of infection can be traced. It is also called as PCR or RT-PCR test. Sensitivity of molecular test is in the range 71 to 98% and sensitivity 95 to 99%. Second type is antibody test. These tests detect antibodies Y-shaped molecules made by the immune response to disable a virus or mark it for destruction. Antibodies are detected in a blood sample. The test measures whether the antibodies bind to the novel coronavirus. This test identifies people who may have immunity and whose antibodies could be used to treat COVID-19 patients. Test can be done at community clinics and blood banks. It shows you were infected by novel coronavirus in the past. It doesn't measure ability of immunity system to fight against coronavirus. Antibody test shows high sensitivity, 96 to 100%, and specificity, 99%, compared with RT-PCR test. Third type of test is antigen test. This is the newest one and detects antigens. It means, it detects pieces of virus that the immune system recognizes. A single virus has many antigen. Infected cells collected on nasal swab. Antibody that binds specifically to virus antigen produce high signal. It provides same information as a RT-PCR test. Antigen tests are rapid and offered in the doctor's office. It shows person is infected now. It shows 80% sensitivity and 100% specificity to COVID-19. A positive RT-PCR and a negative antigen test shows a person is slowly excreting viral RNA from the body and not capable to transmit disease. In serious cases, SARS-CoV-2 lands in the lungs and can do deep damage. The body's response to viral infection can injure many other organs. Scientists are just beginning to probe the scope and nature of that harm. 1. Lungs, immune cells crowding inflamed alveoli and air sacs, walls break down diminishing oxygen uptake. 2. Heart and blood vessels, infection promote blood clot, heart attack, and cardiac inflammation. 3. Brain, some COVID-19 patients have strokes, seizures, confusion, and brain inflammation. 4. Eyes, conjunctivitis, inflammation of the membrane that lines the front of the eye and inner eyelid. 5. Nose, virus may move up the nose's nerve endings and damage cells. Some patients lose their sense of smell. 6. Liver, an immune system in overdrive and drugs given to fight the virus may be causing the damage. 7. Kidneys, virus may attack the kidneys directly, or kidney failure may be part of whole body events. 8. Intestines, infect the lower GI tract which is rich in ACE2 receptors, causing diarrhea in approximate 20% or more patients. The SARS-2 virus is believed to enter GI through the nasal mucosa or the gastrointestinal tract due to their higher expression of protein ACE2. The intriguing part though is that recently reported studies have noted altered mental health in some COVID-19 patients showing symptoms like anosmia and ageusia, thereby indicating a neuroinvasive nature of the virus. CT and MRI scan of SARS-2 positive patients showed acute necrotizing encephalopathy which results in seizures, liver problem, and mental disorientation. Severe neuroinflammation resulting from cytokine storm mainly due to production of interleukin-6 secreted by the macrophages. The systemic inflammation causes severe encephalopathy in the patient, and that may lead even to brain hemorrhage and stroke. Other risk factors that exacerbate neurological problems include as smoking, age, genetic susceptibility, mutation in viral strain, and individual's health or previous medical complications. Myocardial injury from SARS-2 can be explained by two mechanisms. First, myocardial injury can result from the associated cytokine storm manifested by elevated levels of interleukin-6, IL-6, and second, myocardial dysfunction from the direct effect of virus on the heart via ACE2 receptors. Increase in D-dimer level in the blood shows rapid coagulation of blood. Increase in ferritin and LDH enzymes shows liver and cell damage. Post-mortem unfixed lung specimen was moderately heavy, edematous, and relatively firm with area of hemorrhage in the right upper/middle lobes and left lower lobe. Inadequate oxygen supply to the heart due to lung damage increase risks of cardiac complications. SARS-2 infection leads to myocardial injury, myocarditis, infraction, heart failure, cardiomyopathy, arrhythmias, cardiac arrest, and venous thromboembolic event. Risk factors such as older age, diabetes, systemic inflammation, coagulation abnormalities, multi-organ dysfunction, and immobility increase cardiac complications. The number of different blood cells, including leukocytes, lymphocytes, neutrophils, platelets, and hemoglobins, undergo changes that can be evidence for the type and severity of the disease. Many SARS-2 patients showed increased level of blood biomarkers such as C-reactive protein, alanine aminotransferase, aspartate aminotransferase, creatinine, creatine kinase, IgG, and IgM.